present it, to have discussion about common issues, and so even if you don't have anything to present, if you have some topic, some issue that you would like to discuss, just at some point bring up the question, and then afterwards we'll talk about this EMBRC call and possible collaborations within that. So maybe do you want to start? Just as an idea, who has who has something to present? But I heard you had 4 presentations. Okay, perfect.
ahora tienes que escribir. Dale aquí a Antera. Vale. Antera. Punto. Mira aquí. Antera Martel. Espérate porque Antera punto Martel arroba es este. Le da ahí unirse. Okay. Ahora vente aquí. Ahora que se conecte y ya compartes la pantalla aquí. Vente. Sí. Con el tuyo.
Compartes ahí y manejas de...
Sí. No, y lo maneja desde aquí. Vente con el ordenador. Vale. Vale, y ahora comparte pantalla. Lo tiene en inglés. Non down, dile de non down. Lo vas a hablar tú. Y sí. Sí, ahora quitas la cámara. Vale, y ahora vete al FileMaker. Así también. Quítalo el micro. Vete al FileMaker. Vale, ya ven, ahora ponlo en grande. Ese no, este. La pantalla, aquí. El verde. ¿Está? Ya que se mira a ver si se ve en grande.
Eso, pica en gente para que se vaya en gente, sí, para que se vaya al lateral y ahí está.
Vale, perfecto, ahí lo tienes.
Sí, y ya lo tengo yo insertado en este. ¿Cuál es el tema? ¿Cómo el tema que vamos a hacer
la base de datos? Well, I think maybe you can see it here is I don't know if you can see it from there. The structure of the database is divided in the data related with the strains, which is the part we call collection. The ID is identification, mainly morphological and genetic identification. And then we have a part for biotechnological data, mainly how we grow the algae, we follow the growth, and we keep the data there. And also we have services, how it's more related with the services that we do for projects or clients. And also for methodology, consumables, and the equipments that we have in the whole BIA collection, and also biotechnology. So if we go into the strains, we can select, for example, this one. This is a native strain. It's Arthrospira platensis, collected in Fuerteventura in a reservoir. And we have the BIA code, and B because it contains bacteria. and the class, genus, species, and the person who is taking care of this strain, if it is in the public culture collection or not, which is one it is in the catalog, the media, the transfer interval, where it is located in the collection, and some of the data. If we have some other cultures or containers different from the one we use normally, we can express it here. And also if we have deposited in another collections, we can also put the relatives, I mean the code of the other collections here. And mainly for the macroalgae, we have open new fields like the life cycle, etc. And we also have a part here where we can put the data for the collection of the material in the field. And this is the overview. So we have a number of the inventory of this material, where the country, the location, the geographical coordinates, and where, if it is brackish, terrestrial or marine, the collectors, and more data on the location, more in details where it was collected, some pictures. Then we go back to strains. We also have isolation data, who did it, when, which was the method, if different from the one we use normally, some remarks about maintenance. We also have the taxonomy data. For example, we are using algae-based identification for whole, and now we are applying the worms. We will do it as you showed yesterday, which is fine, and also we can do it for algae-based also. And some other data, if this culture has been selected, we call venta, somewhere like this. And if we have give it to some other colleague inside the BEA or the university for doing experiments, so we can do it here. This we can document it there. So we also have the molecular database, the sequence and the region has been analyzed, and the access number in the gene bank. If we go in here for the sequence, we can have the result of the blast, and we can do the blast from here and also check directly in algae base. We also have the morphological characteristic here done with for a person who was a taxonomist at the time. So if he checked the morphological characteristics, and then we consult or get into discussions with the genetic results. Not in this case, we was very clear that it was Arthrospira. Then we go back to strains, and if we go, for example, to the Biotech, we can access to these modules. This is a kind of general screen, but we can go to these modules one by one, like DNA lab, multi-tax sequence, or MOTAX, or morphological taxonomy. We have also for a photo gallery that it has been, we have to work very hard on that. And for biotechnology, for example, we have, let's check one of the. So we have a culture here that was done for a project called Sanobio Arthrospira, and those are the data for the culture. Maybe we can go in. Let me see, one of the Savannah project, for example. Nope. Okay. Then we have 8 cultures for this project, and then we can go into one of them. And here you have, we can even express the data of the growth curve and so on in here. You can deposit here the Excel files or Word file or PDF with this data. We can also see the curve in here if we express them, of course. And then we have the collection. When we collect the biomass, and how many grams we have, and also now we are implementing the biochemical. Let's see, go back to here, and this is the biochemistry. Those are the data that should be implemented. Now we have the format, and then we will go ahead on, and we will have all the process from the sample that was collected, the sample that was isolated and maintained, the sample that has been grown up, on the data of the growth curve, how much biomass and in which conditions we get this biomass, and the data of the biochemical composition. So this is not ready yet, but it's coming. And we also have here, for example, methodology. This is most likely implemented for the ISO, so we are on the way also. And then we have, for example, how we prepare seawater, enriched seawater. We also have here a list of equipments or consumables. And we also have another, but different, in a different place in the server for administrative work. So we have the contracts, the projects, how we manage administratively the money, how we split it, and how we do we have the los gastos.
How you spend the money?
Yeah, the spendings, so to say. So everything is, in this case, this part of the database is shared with the administrative department. So this is not the one I can show it right now, because it's more administrative. But we are building this just because it has been growing up depending on our necessities. So also for cytometry, if we pass some samples for cytometry, we have this, we can register it here. for cryopreservation also. See. Okay, how we did it, the method, when, who, where it is in the minus 150, when we did the viability test, and so on. So the idea is to have a place where to register all the things that we do with the strain, to get the more traceable method or more traceable data that as we can from our work. So any question? But this is a FileMaker program that has been built up from our idea on how to work or how to express the data. In the beginning was to fulfilling the GBIF idea of having the data as much as detailed for being comparable with another collections. So, but it's in constantly changing and growing, so to say.
Okay, Antela, just remember that we have the possibility to prepare a specific output and send the information, for example, to an Excel file. I'm thinking that the discussions we have yesterday for sending to EMBRC the data of the strains. So it will be quite simple to take the marine strains, for example, as a first group to prepare the Excel file the way you show us, and we will send all the information.
Yeah, we can check public, BIA strain, because we have EDA, which is those strains that are maintained for patents, and other that we maintain for research, just for compare with our cultures. So we select BIA 1, which is that is in public strain, and marine. So it will list a list of strains that we can express in CBS, and export to the web page.
So thanks for the presentation. It's nice. It looks like a nice system. It's almost more like a cross with a LIMS, in fact. LIMS system. It's a laboratory information management system. So File Sender is a commercial product. So how much do you pay for using this?
We pay for the license of having FileMaker. But it's paid by the foundation because it's the system that they use for the whole administration.
Okay.
So, and then we we have a blank sheet, and we build it up, saying I want to have this, and then just you create it from.
So it's an online system only.
Yes, it's an online system.
And presumably has backups and things like that?
Yeah, we have several backups in the server.
Mm.
Then this is for those computers that has the license. But for the new catalog, we will have it online for the fields that we decide to show up. So people can go into the FileMaker through online from their houses or work and check our go into this.
So the users can go.
The users will do it. That's what I told yesterday about having automatic input. So if I change something now here, they will see it just in the same moment. Because now we have to create to generate a list of the cultures and to produce a CSV and go to the web page and incorporate this.
And that is done automatically or manually?
No, I have to do it.
Is there no way in this system where you can automatically generate a CSV?
Well, no, I have to do it myself. It's let's see.
I mean, did you look into whether it is possible to do it, or it might be possible to do it automatically? I don't know.
I hope so. This is export and import, and then I can export and import the data that I select, and then I can do it in Excel file, a PDF file, or CSV
it's
file.
for the question of.
But I have to do it myself.
Yeah, because it would be the same for trace. Basically, you would have to. But I don't know with this system whether you can set up an automatic thing or not.
Probably. Yeah.
And so who is the kind? It's someone in the university who manages the system?
No, it's an external person. IT. Yeah.
Okay, so we would need to.
I can go into, because I'm administrator, and do some changes here to just open some fields, change some. For example, if it is a number and I wanted to have a number and text, I can change some small details, but I'm a little bit.
And can you change the graphic design of it, or?
Yes.
So on the website you can make it look like a nice.
Yeah, it will be not that way. It will be harmonized with the rest of the web page, because we have instructions from the university, and we have to have certain colors and size of the letters and so on. So it will be shown up with the design, but behind that is this database.
And another question, so your taxonomy, you say it's from algae base?
The algae base, yeah.
Yeah, is it?
This is the for the.
Is it automatic?
No,
So
that was one of the new implementation, because we have done it, or we are doing it with Paula. One by one, and then our IT discovered what you showed us yesterday, also for worms and also for
so you can
algae base. So we will do it.
Yeah, okay. That makes it much easier, and it makes
easier, and we could see that some errors when you write the numbers, the names.
Yeah.
So. We started with an Excel file, so and then we developed this for us it's more practical, while you know how it functions.
The important thing here is that we can improve it from time to time, as far as we see things that we want to come into the database, we can do it, and it's very nice to work with it.
Okay, thank you.
Okay, thanks.
Next, volunteer.
Oh,
You have the presentation here.
yeah. So,
Is this okay? Finger? Yes.
yeah, I didn't know what was expected to be presented here. Yes, thank you. So I just wanted to give you a glimpse of the Marbach activity, just to kind of showcase the diversity of the collections in the EMBRC, and say a few words just about our latest focus. So we are up north in the Arctic oceans and sub-Arctic and sub-Arctic areas, and here you can see the sampling area for the Marbach Collection, and we have been collecting most of these samples ourselves. So it's close to 1,500 locations, ranging from the southwestern part of Norway and the fjords down there up to north of Spitsbergen and Svalbard, as you can see. And so this is not going to be much about microalgae and databasing. It's Marbach, we started out with the invertebrates, and today I guess we have a total of 800 different species that we have been processing, because these, I will show you on the next slide, these are finite samples, so they cannot be cultivated by us at least. So they are collected offshore and brought to the lab and processed, and then preserved, kept in the freezer when they have been preserved into processed into extracts. And then the next thing we started up with was the bacteria, and the latest focus here has been actinobacteria. So I guess there are a few thousand isolates there, and everything from, as I said, up north. It's not quite correct to put up microalgae here, because in fact we don't have it in the physically in the collection now. So it's a close cooperation with the University of Tromsø, the Arctic University of Norway, and it's they have been doing a lot of interesting work and has a great kind of showcase when it comes to cultivation of Arctic diatoms. They have a cooperation with a industry plant outside of Tromsø, where they are cultivating these Arctic diatoms by using harmful CO2 and NOx gases from the factory smoke, and that comes from ferrosilicon production. And at present they've got the world's largest vertical column photobioreactor. It's 300,000 liters, so it's quite successful. And they can, what's special with this Arctic diatoms is that they are using it, it can produce a large amount of biomass before the water is colored, so it's good, really good light penetration. So, and thinking about the situation up in the north, we have a huge algal bloom in the springtime, and light 24 hours a day, you know. So it's quite special. So as far as I've understood, it's these strategies of living of these Arctic diatoms that is kind of the background for the setup at the fabric plant up north. So and they are producing well, first and mostly I guess it's for fish feed, biomass is for fish feed, but they're also thinking of biofuels, and there are interesting enzymes, and they have a close cooperation with other research groups at the university. So bioprospecting is also in focus, and looking for secondary metabolized produced under different conditions here. Yeah. Okay. And the database. I didn't bring a more detailed presentation here, but what's important for us is that we have got this offline module of this online system. So we bring that with us in the field and on cruises. So that's quite essential for our activity to be able to do that. Yeah. So as I guess this extracts of microorganisms is essential for us, and is the species, in fact, where the pharmaceutical leads has gone the longest way in this value chain. So the most interesting finding is from an Arctic species. I think it's a species like that. I'm a chemist myself, but so no, it's they have found molecules that kind of is kinase inhibitors, and is going on to the first clinical studies from these findings. So, but preparing extracts from microorganisms, as I said, it's finite samples, so it's kind of a process where we are out there in the sea collecting them, and we had this taxonomy analysis, of course, and in the database it's linked to ITIS. We want it also to be linked to worms because it's a better one. And we are processing, and as you can see from the organisms out in the ocean, on the seafloor, and at the lab, it looks like these aqueous and organic extracts. That's the product out to the users of the samples. So, and then lately we have also been focusing a lot on marine fungi. And what's that? Well, it's just fungi that are found in the ocean, and you can find them on wooden structures or on driftwood as free floating cells in the water, and infecting other organisms or breaking down dead organic material, and they even form symbiotic relations with, for example, microalgae. And there are only about 1,500 marine fungi species described in the world, and only a few have been found in Norway. It's about 20 to 30 species. So they are generally not well studied at all, and it's considered a fairly new field. And in Marbach we have Ole Christian, who's a specialist on this, so I guess the competence in Norway is very well presented in Marbach. And the workflow here is, the method is to isolate fungi. Well, it depends on the substrate we work with and the type of fungi, but generally we plate them out on agars containing antibiotics to prevent bacterial contamination, and when they are considered pure cultures, they are characterized by morphologic help me morphology and a DNA sequence. Sorry about that. But and then, in order to preserve the cultures, we submerge agar pieces in 20% glycerol before storing it in biofreezers. And there's a lot remaining to explore regarding marine fungi. At Marbach, we are working on describing, in fact, several new marine fungal species from colder Arctic waters, and it is, for these and others, expected that they have novel enzymes and chemical molecules, secondary metabolites, that can be used in different industrial applications. And the last few years there has been more new compounds isolated from marine fungi than from marine bacteria, as you can see from the illustration here. And the marine fungi, the ecological functions are poorly understood, and they might impact different ecosystem processes. Impact the ecosystems more than expected. So it's interesting to work with, and it's just, as I said, a part of the Marbach Collection. Well, this was just showing the diversity of the EMBRC collections and what Marbach is focusing on. So it's not that much on algae as has been presented here, but it's just to give you an overview on what we are focusing on. So thank you. Yeah.
Okay, it's very interesting. About the fungi, you also grow then in fermenters and grow to get biomass and make extracts and this kind of things?
Yes, users of the samples from Marbach. What I presented in the big photobioreactors was users of the Marbach material, so it was a cooperation with the University of Tromsø. But that is the way you do it. It can be cultivated as many others, other microorganisms, yes.
And at the collection you have, you can keep the material just cryopreserved or freeze-dried?
It's cryopreserved. I have to look at Eivor as well because she's a microbiologist. but it's quite easy and quite interesting, yes.
There's something I don't quite understand. So in your collection, it's just extracts or it's living strains as well?
For the invertebrates, it's just extracts.
But for the fungi, you have the living strains?
Yes, we cultivate them.
And they are available via EMBRC and?
Yes, they should be. Yeah, in the end. So it's only the invertebrates that are these biochemical extracts and the finite samples. And
a question about the extracts of invertebrates or macroalgae as well.
Yes, of course.
How do you clean them before you do the extract? How do you know there's not like epiphytes or other things on the, or do you not bother?
In fact, we do not know and we don't bother because for this, isn't that correct, Eva? Because for the setups, we're just presenting the way we do it. So if there are symbionts or living together here, it's all about looking for new bioactives, you know. So at the end of the day, you might want to know where it comes from. But for us, it's a sample. It's processed like this, and it's collected there, and this is the way we have done it. So we can't be sure. But I guess it's that. The findings that we have that has gone a long way towards the clinical studies, I guess they have checked that it's the invertebrate, the macroorganism itself. But I guess from the Bibles of marine natural products, we know today that, well, most of those that has been reported from invertebrates has been, in fact, produced by a microorganisms that's living in as a symbiont together there. So we don't do that, isn't that correct? Again, Eivor, because it's all about the bioactives.
Okay.
Okay, thank you. Yeah.
Actually, I have a question more focused on cyanobacteria. and I would like to know if you are using whole genome sequencing, and what do you think about this? What's gonna happen? What's the implication of using this whole genome analysis or phylogenies?
I don't know if the question was just for me, but if I can say something about that. I will show in a minute that there are some of the strains in our collection which have genome sequences, but we've just started. We're involved in a very big genome sequencing project. And that's partly why I was asking questions about cleaning invertebrates, in fact. So I think for cyanobacteria, I don't know how many species have actually been done. I know there's a lot of Synechococcus and Prochlorococcus.
About 3,000, I think, something like that.
That have already had a genome sequenced. Yeah. So I think it's quite easy in cyanobacteria. And I think for now, in collections we tend to do barcoding or 1 or 2 genes, but I think it will not be long before everything goes via a genome sequence, and the methods will get easier and easier and cheaper, and I would predict that in 5 years' time, all of us, rather than doing a barcode, we're systematically doing genomes for all of our strains. So.
Okay, so what do you think about the definition of the core genome to phylogenies? And I think people are not analyzing the HGT, and even if it's core genome, horizontal gene transfer exists. So. And I have doubts about the lengths of genes, because longer genes and more variable genes, they will have more influence on phylogenies. So is this good or bad? Because we're gonna have different results from the 16S phylogenies. So what do you think it's going to have? It's a question for everybody. So.
It's for everybody. So if you have an answer, I think it's a really complicated question, actually. I think you're right to say that. More and more you see now multi-gene phylogenies, and it doesn't always give the same results as a 16S. I'm not a specialist of the bioinformatics part of it, so I couldn't tell you which one would be more valid. I could put you in touch with people who could answer that question, but you would think that it will go more towards multi-gene phylogenies, and they will be the ones which would be maybe not with all genes if you have criteria to take out the longer ones and stuff like that. But complicated question. Maybe someone else has a better answer.
I do not have a better answer, but back to, we are also kind of part of this Earth Biogenome project. I don't know whether it was that you was referring to. So out in the field, we have also kind of flash frozen samples of these microorganisms for better studies of the genetic material. But for us, we do whole genome sequences only in collaboration with the users and then samples that we where we have really interesting molecules that we have identified as bioactives. So it's to have just a closer look on what's producing this and so on. So all genetic sequences that we have on the material, we add a link in our database to be able to find them. So that's kind of important for us as well. Yes.
I have another question about the ecology of microalgae or cyanobacteria also, and the methods of isolation and the implication of them. For example, I sample something here in Canarias and strike on a agar plate, and there we're gonna grow 3 or 4 different species, and I isolate one, but this one I cannot find on the nature material. So can you say that this species is from Canarias or not? Okay. Okay, I'm walking through the island and I collect a natural sample and I strike on an agar plate with solid medium. And there we'll grow 3 or 4 species. Okay. I isolate one of them, but this one that I isolate, if I go to the natural sample, I don't find it. Is it from Canarias or not? Not a contaminant, but it might be from a actinate, a spore, or a dry cyanobacteria, or a dry algae that came with the wind or something. It's there, but it's not growing. It's not alive. Maybe it's from somewhere. It came with the wind. So, and what taxonomists and what we are doing, well, I don't like to do this. I don't do this. But many people do this and attribute the habitat and the place of the new genera, new species they are describing based on that. So they would say this is from Canarias, from a dry environment, and that's it. So what do you think about this? So this is. Do you think that this species is from Canarias, or can you be sure of that?
Sounds like another question for the bar later. I mean, I never really thought about that, but yes, I would say it obviously comes from Canarias. It was in your sample, it was alive. It may not have been active, but it was alive, so.
Okay, but again, if it is only an actinate that came with the wind, so it's not alive, it's there, but so it's not growing.
If it's grown back, it's alive.
It's not growing. It's not the typical environment. Maybe it's from the tropical forest. Maybe it is growing very well in a tropical forest, but.
And does the. It's an interesting existential question, but does it have an impact on something? The answer, or?
Sure, because it has impact in taxonomy and the ecology of the species and genera. Yes. Yeah.
Because just because you don't see it in your sample when you go back to it doesn't mean it's not there.
No, but it's possible.
Recently, I read about this in the scientific articles. They talk about the suggest that the microalgae are cosmopolitan because it's the proportionally in relation to the size of the organisms and the distribution in the world. When the size are small of the organisms, they are in all the world.
Yes,
You
that's the
understand?
everything is everywhere theory, but this is applies for some, but not for all microalgae, I think, or not all cyanobacteria. Yeah.
I don't understand you.
No, this is possible that everything is everywhere. Yes, but sometimes for some species or for some genera, this is not true.
Yes, it's 75% of the species are cosmopolitan.
I really don't know what to say, but we have a lot of dust from the desert, and we have trapped many microalgae. We don't know if coming from Africa or they are from south, that it comes to north. But of course, and sometimes they grow in, for example, some chlorellas vulgaris that they are identified as freshwater algae, and they happen to grow in the marine. So, or find it in salt work. So, what to do with this? Because you check the taxonomy related to the description on how where they grow, they cannot be a chlorella vulgaris. So something is wrong. It's wrong the algae that it was adapted there because of the winds. They come from some other place. Or it's just we have to do a whole genome and find something strange there, find it in a cotton or so. So there are some cryptic cases that should be going too deeper, but it's quite tricky.
In terms of ABS, it's where you sample it, so it would come from. We had that question with a microbiome project, and they were talking about the ABS of human gut microbiome, and so it's where you sample it. So your microbiome here belongs to Spain, actually. Even if you ate something in another country and then come here. Okay, so I'll just talk a little bit about. I just wanted to talk a little bit about a few projects that we have at the moment, just to give you a kind of idea of the kind of things we're working on at the moment. So the first one is a French national ANR, is the National Research Agency project. And this is a kind of a taxonomy project called PhenoMap, and it's the phenotype gap for marine phytoplankton. So it's us and then some people in Ifremer. And this is results from environmental metabarcoding. This is mainly Tara Ocean. I don't know if you know of the Tara Ocean campaign, where they went around the world and sampled and did metabarcoding of environmental data. And this is results for phytoplankton. And what you can see, so here is 100% match to a reference sequence. So 100% identity to a reference sequence is the white, going down to 90%. And what you can see and it's ordered by the most reads that were there. So Dinophyceae was the most abundant, and etc. But what you can see about just about everywhere is that there's only like, I don't know, 25% which actually matches a reference sequence, and all of the rest of this going down to like 90, well, for a start, match to a reference sequence from GenBank. But then when you actually go into GenBank and have a look at the reference sequences, and this is what we've done for some phytoplankton groups, we went in and kind of had a look for some groups whether the reference sequences could actually be trusted. And obviously you can trust it if it comes from a type strain. That's the obvious thing you can trust, because that's obviously the reference. But there's a lot of other cases of people who have published a sequence, said it is such and such a species, and sometimes there is some morphological evidence to back up the fact. But in many cases, there's no evidence at all that what they actually say is the sequence of this species is that species. And it takes a long time to go in and look at all of that. But when you do it, that's just an example for one group, the Pelagophyceae. You see there are quite a lot of the reference sequences for pelagophytes. There's no actual evidence that the sequence is really that. So even for the reference sequences, you can't really trust them all, unless you go and have a close look into the thing. And then all of the rest of this part, and I say going down to 90%, this is 18S data, so going down to a 90% identity is really quite divergent. And all of the rest of this is we don't know what it is, in fact. And so the idea of this project is to have a look at what these things that we don't know what they are. So to improve the reference databases by linking taxonomy and barcode, so verifying and validating existing reference sequences, which is what I was just explaining, generating reference sequences for as many new species which didn't have a reference sequence, and then describing new species as well when we find them. And we're doing this a few ways, and the most obvious one for a culture collection is to look at the cultures we have. So we've been doing a lot of sequencing of the strains we had, because we had many cultures in the collection, which have some which have been there for a long time, which we'd never sequenced. So we've been doing a lot of sequencing and then microscopy work to characterize these. And we came out with some new things from this, and some of them are really quite different from what was known before. And this is one example of a new class which I collaborated in the description of this new class of algae that we had in the collection, which is in the haptophytes. It's a new class of haptophyte. That's another one actually, which this is the ochrophyta, and there are several environmental lineages. So we know they exist from environmental sequences, but we don't know what they are, and these are called the MOC 1, 2, 3, 4, 5. And it turns out that in the collection we have one of these MOC 5, which is a lineage which is closest to the Eustigmatophyceae, but long branch. And this is just a really boring small flagellate cell which has been hanging around in the collection for 15 years or something. And when we finally actually got around to sequencing, it turns out it's a new class. So there are things you can discover, interesting things even in your collection. which is why you should never throw away a strain, by the way. But a lot of them are really boring because that's just one example. Picochlorum is a small green ball, basically. And there's 5 or 6 species already described, but from the genetic analysis, it looks like there are at least 5 more species, but they all look exactly the same. And so, you know, a lot of the new diversity we're discovering is cryptic, but still, it's interesting to describe this diversity. And that was some time ago, actually. So it's not up to date, but that some time ago we discovered almost 70 new species in the collection, which waiting to be described. And the big work now, and the bottleneck on that, is doing the characterization by electron microscopy. So that's something that, you know, we're working on and happy to collaborate on and stuff like that. So there's a couple of other ways we're looking at this unknown diversity. One of them is using fluorescent in situ hybridization, so fish. So this, when you have the sequence of the environmental sequence, which you don't know what it is, you can design a probe with a fluorescent tag on it, and then go back to your sample, apply this probe, and whichever organisms it attaches to, which is your target organism, will have a fluorescent signal, and then you can try and characterize the morphology of what it is. So yeah, this is not really a culture collection activity because we're not trying to culture them, but it's still, I think, an interesting way to do this. And that's just what the method looks like. And there's several challenges to do this, to doing this, and I won't go into the details. And the other way we're trying to characterize this diversity is doing single cell. So we're not trying to culture them, but we're doing single cell genetics. So we're isolating cells, taking an image of them, and then extracting the DNA from the single cell, and so you have a link between the image and the sequence, and this is working very, very well, actually. And that's just a few examples of some of the crazy organisms that you find, which no one has ever cultured, and I'm not sure they would really be very easy to culture, but you know, this is a way to get around the fact that we can't culture them. So crazy dinoflagellates mainly, you find, I think you'd probably find quite a few of these here, actually. Different dinoflagellates. And coccolithophores as well. There's not any. So these are actually very, very small, and the single cell works very well. So that's a kind of taxonomy project we're working on. And attached, this is something as well which I think might interest some of you. Attached to this project, we're obviously depositing sequences. We're actually very involved in the RCC in the PR2 database, which was developed in Roscoff, which is a reference database for, it's a curated reference database for barcodes. And we're kind of supporting that resource. And then there was also this idea to, you all know, I guess, the Red Book, the Carmelo Thomas book, which is now 30 years, 20 years at least old, and it's getting a bit outdated. So our idea is to see if we can, well, our original idea was to see if we could put this book in a online form and try to then update it. And Carmelo was very happy for us to do this, but unfortunately Elsevier were not happy. So we're actually in the process of trying to develop an online kind of taxonomy identification resource using this microtax system, which is a system which was developed by a colleague working on coccolithophores. So it's a system where you can put images and taxonomic information online, so it's actually very, very good for coccolithophores taxonomy if you're looking to identify coccolithophores. But we're looking now to try to extend this to other groups. We started on dinoflagellates, and so if anyone wants to contribute to that effort, because it's an enormous effort, and we'd be very happy to have contributors to that. So just a few words on a couple of other projects. So this is the project that I was just talking about, the genome sequencing project. So it's called Atlasy, which is just starting right now, and it's a French national project with the objective of sequencing 4,500 marine eukaryotic organisms from French waters in the next 6 years. And so it's part of the ERGA, which I'm not quite sure what that stands for. It's the kind of European genome initiative. It's big funding for this project, and our role in this project will be to provide strains. And so we've got around 900 different species from French waters for this. So we have we're in the kind of phase of trying to develop the protocols. So, I mean, it was an interesting question. I have lots of questions about how we're going to provide what do we need to provide in terms of quantity of biomass and or DNA for the sequencing. Are we going to need to axonize the cultures? I don't know if Martin wants to axonize 900 strains. That would be. So any experience on that kind of thing, I'd be interested to hear about. And that's why I was asking as well for are you cleaning the macroorganisms, because this is not just microorganisms, this project, it's also invertebrates and things. But obviously if you're doing a genome sequence, it kind of has to be clean. And I'm not quite sure how we go about cleaning the invertebrates for the moment, but let's see. And then just a couple of European projects that we're involved in, which are just both starting out, and they're quite similar projects, and these are both projects on microbiome research. Microbiomes are very fashionable at the moment, and so we're involved in these two projects. The blue remediomics, I won't go into the details, but it's basically to take marine microbiome samples, but to develop this kind of discovery platform with a lot of focus on using the bioinformatic data, so the sequence data that already exists, mining that data to find activities, to find core microbiomes and things like that. And then we're involved in the culturing part, and then there's a screening part with different applications. So I won't go into the details for the moment. And the micro project is very similar. It's very similar, but not just on marine microbiomes. It's on marine microbiome, human seeds, and soil, and things like that. So, yeah. And the kind of things we're thinking of doing, or we're planning to do in these projects. So we do a lot of cryopreservation for the last more than 10 years. That's just the standard procedure we use, 5% DMSO, 1 degree a minute in a controlled rate freezer, then to liquid nitrogen, then storing in minus 150, and we have around 2,000, something a bit more maybe, cryopreserved at the moment. So that's something we do in routine on our collection, but and that's just shows that's also a bit outdated, but it shows the different groups, which you probably probably can't see, but it shows which proportion of different groups are successfully cryopreserved. A lot of cyanobacteria, it works very well. Diatoms and green algae and things like that. And some of the Perkinsia, they are parasites, it works very well for those. So what we're planning to do in this project is actually to see if we can cryopreserve natural communities. And kind of the objective of this is to see if we can, while we're on sampling missions on boats or something, that you can now get these portable cryopreservation devices. So the idea is to see if we can cryopreserve samples, mixed sample, natural samples, while we're in the field, and then bring them back, and then you can thaw them out whenever you're ready to then do isolation afterwards. So we've started doing some tests. It's using different cryoprotective agents. I think there's more of them than that. I think there were 7 different things, and then looking with controls, obviously, and then looking after thawing them out the day after, and then a few weeks after, and seeing if we can do RNA barcoding to assess, because if you just do DNA barcoding, if things are dead, they still have their DNA, so you wouldn't know who was still alive after the cryopreservation. So we'll do flow cytometry and RNA barcoding to see if we can develop this method. I think it would be a nice, because you don't always have the time or the possibility to do the isolation straight away, so it could be quite a nice thing for that. And the other aspect we're looking at is microbiomes. So all of us who have microalgae cultures, unless they're axenic, we actually also have lots and lots and lots of bacterial species in our collections. So we've started in collaboration with some partners in the project, looking at the microbiomes of some of our strains, and the details doesn't matter, but this was from 60 strains, and it was just 4 genera. So I think there was Phyacystis and Micromonas, and so just 4, we picked 4 genera with like 15 or 20 strains each. And these are the amount of bacteria we have in the collection, and the relative abundance. And so you can see most of the strains have at least 15 or between 10 and 20 bacterial species in there. So this is something that we don't think about that much, but something that we want to go and look at. And the kind of things we're looking to do is co-culture experiments, and particularly this project is also based on looking for screening for certain activities or metabolites. So what we want to do is take some strains which produce an activity or a metabolite of interest, determine the composition of the microbiome, so sequence the microbiome, then try to isolate the bacteria into monospecific culture. So we have the bacteria as monospecific cultures, try to axonize the eukaryote, and then do co-culture experiments using the eukaryote with each bacteria individually or mixtures of bacteria, to try to see which of the bacteria are having an effect on the production of on the growth or the production of the metabolite of interest. And for me, this is a project which is focused on I'd say biotechnology, but the real interest for me is seeing if we can find a kind of miracle combination of bacteria that we could then add into cultures to make them grow better. Or I think potentially when we isolate new cultures, this might be interesting to have a kind of bacterial boost that you can put in when you isolate, because there's a lot of things that we isolate and never grow. So maybe this could be a way to improve the success of isolation and things like that. So for that, I didn't find the photo of the, we're using some kind of glass co-culture chambers, so which are between 100 and 500 milliliters, which have a membrane between the two. So you put a 0.2 membrane or 0.1, and you grow the bacteria on one side and the axenic eukaryote on the other. So I didn't find the photo that was supposed to be there. But we're also probably going to look into these. There's quite a few publications recently which are developing 3D printed co-culturing devices like these kind of chambers. You can 3D print them and have either just two of the wells linked or several wells linked and things like that. So we're going to look at doing that. So that's it. That was just a few things that we're working on at the moment.
Did you try to do that single cell sequencing for species with mucilage?
Probably not, no. I mean, the majority of what we've done for the moment are open ocean pelagic species, which tend to not be so much with mucilage. But I mean, I know that is an issue, even when we're trying, we have some cultures of more benthic microalgae which produce so much mucilage that it's becomes like the cultures become like a shower gel or something. They, you know, and those ones we have real trouble to extract DNA and stuff. And I think there are some methods for reducing. Do you know what you would add to reduce the? Sorry? Darkness. But is there not a chemical treatment for reducing mucilage? I think there are some chemical treatments for that. I can have a look into that if you want. Because you have an issue with that?
Because of in cyanobacteria, you know, Stigonema, they don't grow in culture, and they have a lot of mucilage, so it's very hard to sequence.
I can have a look into that. One of the big drawbacks or the limitations of this single cell sequencing is that the method we're using, you only get a light microscope image. So we're using inverted light microscope. So you isolate the cell on an inverted microscope, take an image, a light microscope image, and then put the cell into the buffer. And for a lot of things, and particularly the really small things, you can't get a good taxonomic identification from a light microscope image. So we're looking to see if we can find a way to do scanning electron microscopy before you extract the DNA. But it's something we're trying to work out. I've only really succeeded in blowing up our SEM for the moment. But so I don't know, I say that because I don't know, with cyanobacteria on a light microscope image, can you get an accurate identification for species?
Yeah, thank you, Ian. In relation to the first project you saw us about the sampling around the world, do you remember, well, what kind of sampling? Is just surface waters?
So the Tara expedition, if I remember correctly, they did surface and DCM.
Okay.
Yeah, and actually you can go into that data and look much more closely at which ones are surface and which ones are DCM.
This is more or less related with the question before. That well, you know, we have here a very big discussion about what is a native strain of microalgae in our surrounding waters. Our idea is that all the microalgae are ubiquitous, and it's just the luck you are when you are sampling. Right, so from a cruise like that, I don't know how many strains are repeating around the whole sampling task. You know what I mean?
Yeah, I mean, I think I don't have the answer like that, but I think that analysis has been done. The thing is that the Tara project started in 2012 or something, and at the time, and it's still kind of the case, you're using an 18S barcode, but only the V4 or V9 region. So it's only, I don't even think they're 200 base pair. You know, it's very short sequences, which means that you get much less taxonomic resolution, which makes it even more surprising that you get such a you know, so many yeah. But so I'm not sure whether that would give enough resolution to, you know, look at microdiversity and see whether you have a localized population of certain species or.
They are moving around.
Yeah. But I would tend to think that data does show that there are some species which are really fairly ubiquitous.
I have a questions for all the collections that perform cryopreservation. What kind of viability tests you perform and when? Periodically, annually? How you manage that?
Well, the microphone, so I can answer for us. So, except unless we're doing some specific experiment, our viability test is basically wait for a few weeks and see if it grows. And it's like yes or no. So we don't do a specific
just
quantification. No, it's just does it grow back within a certain time. And I would say that for the ones where it does grow back, so we do always do a couple of extra tubes which we're going to use to test whether it's successful. Your question is, do we then go and check again in a year's time or 10 years' time? No.
Okay.
No.
And all the strains you cryopreserve are xenic or not?
No.
You don't have, before you unfreeze, you don't have problems with the bacteria or other stuff?
Yes, yeah, obviously the bacteria love cryopreservation and come back strong, but you know, for the successful ones, the microalgae do get going eventually. But yeah, it is an issue. So, but so you're suggesting we should axonize everything before? And
No. I'm asking because I have the same problems, so I see if someone has new ideas to deal with this.
No, I mean I think it would be, ideally we would do a real viability test and quantification, but it's just a question of if you do that, you have less time to do more strains. So it's really we've had somebody working on that for what, half of their time, a bit less, for the last 6, 7, 8 years. And we're only up to 2,000, and we're isolating much more quickly than we're cryopreserving. So for us, the strategy is really to try and get as many done as possible, and even if that means that you don't do a real serious viability test.
Okay, thank you.
Okay, we have some axenic ones, especially those that are deposited because of their type strain, so they are axenic, but mainly they are not, and we wait for several days to check viability. That is only to see if they grow or not. We do not do this complicated viability test and counting and so on. No.
Anyone else have a No. Any other questions? Anybody else have anything to present, discuss? I was led to believe. Is there a coffee break? Yes, I think it's probably a good time for coffee. When we come back, we'll talk about these the possible collaboration projects. EMBRC call for projects, and I'll just give you the details. I just have to. So have you heard about this already? So the call for joint development activities. Okay, so there is just published, I got the information through the French node, not directly, but so there is a call for EMBRC is going to fund joint development activities. So what do they say? It's the aim of this call is to encourage interactions amongst our nodes in order to develop new services, which will be available for users of our scientific communities. The call is internal, so only for EMBRC partners. There is no restriction in the subject of the JDA proposal. However, the proposal should be relevant to the EMBRC mission and EMBRC science strategy. So there's a document about the science strategy, which we could talk about after. So there's a call, it must be on the EMBRC site. There's the call where you apply. Aim of the call: develop a new service to be included in the EMBRC service offer. Who can apply? Teams of scientists and/or technicians affiliated to EMBRC partners. There's a procedure. Timeline for submission of proposals from May the 9th until September the 4th this year. Each proposal will be evaluated by 2 GA members based on different countries from those of the JDA teams. So it won't be our people, evaluating. Evaluation criteria: description of the JDA activities, contribution of each operator to the different activities, assessment of JDA needs and its impact, prospective outcome of the JDA as a EMBRC service, link to the EMBRC science strategy, and budget justification. So timeline for implementation and final delivery up to 2 years. Blah, blah, blah. Budget 100,000 euros or 35,000 in case the awarded team is affiliated only to one partner. I don't quite see how you can do a joint development activity if there's only one partner, but anyway, so it's up to 100,000 euros. Eligible expenses: personnel, consumables, small equipment, travel. So there's this call. It seems like a really good opportunity for this working group to get some collaborations and do some interesting stuff together. So who has ideas? Nobody? Well, I can start. I have a few ideas, but somebody must have some ideas of services we can provide. So what I was thinking about, so I think technically speaking, if we bring new organisms into culture, or groups of organisms, shall we say, I think we could sell that as a new service. And what I'm thinking is, for one, in terms of animals, I know there's not really anyone here who is involved in that, but I think I would like to look into the possibility of doing a project for developing new model invertebrates, and particularly I'm thinking of ascidians, because there's some interesting new species. There are a couple of ascidian model species, but there are some interesting new ones which we could develop with some work on them. I think the same could go for macroalgae, and I don't have a specific target, but I think we could probably, for the people who are involved in macroalgae culture, we could probably, have a set of target new organisms which are not actually in culture at the moment, and collaborate to try and get these into culture.
Yeah, involving new techniques and protocols to keep alive material as a collection for macroalgae.
Yeah, for example.
Yeah.
And for microorganisms, what I was thinking about, and it was interesting to see your presentation in Norway. What I was thinking about is that what I thought was really missing in EMBRC collections is fungi. So I thought we could maybe, do a project on, increasing the amount of fungi which we have in culture and which are in the EMBRC service offer. But so that's a few ideas from me. It can also be obviously other types of services. So you could think about, I don't know, taxonomy service or cytometry or such stuff, but in a sense some of those things exist already in some partners, so I don't know if that would count as a new service. So for example, I think you do as well, we have a service for identification, so I don't think we could really sell that, but so that's a few ideas. Anyone else? Any comments? Any? Sounds like everyone should be going to the bar now, isn't it? So no one has any comments or ideas, or maybe you don't want to get involved in this, or training courses. I mean, that's something I was thinking we can talk about that now, in fact. So in terms of, we had that as an item on the agenda, in terms of training courses, maybe we can just do a quick survey of who actually at the moment offers training courses. Or so for us in Roscoff, we offer, and it's been the last couple of years, we offer a training course in basic microalgal identification and culturing, which is a 1-week course, which this year we were actually splitting into 2 courses, one which is identification, one which is culturing. So that's something we've offered for a while. We're also involved in, or some of us are involved in some and teaching on some other courses which we don't organize ourselves, but on microalgal taxonomy identification. So I teach on a course in the States, and a couple of years ago we organized the advanced phytoplankton course in Roscoff, which is a 3-week intensive taxonomy, marine phytoplankton taxonomy course, and the next edition of that is going to be held in Naples next year, I think. Do we have any other training courses? Is that a question? You're asking for it? Yeah, I mean, in our identification courses, in all of those courses, in fact, we do a short introduction to molecular techniques, but it's not, you don't have time to do a detailed. But I know in Roscoff, and I guess in several other institutes, there are training courses organized by well, I guess more on bioinformatic treatment of sequences, and not so much on the actual methods, I guess. I don't know if there is a kind of the actual extraction and things like that, if there's something like that. So any other?
Because of money, can we think about a project to be closer to the companies which need us, or
More specifically?
I don't know, it's something that we could integrate the private companies in the project, and we
You're talking about these JDAs?
yeah, it'd be more close to them and exchange more things and knowledge.
I don't think you couldn't fund a private company, but the whole thing is, are you developing a new service? I mean, private companies are users of our service, so I guess if you have an idea, you could and with for a service which would be of interest to a company, you could have a project where you develop that service in collaboration with that company, and I think that would be feasible. Yeah. Just back to the training aspect. Do you run any training courses in?
We are thinking in the end of the year to create a course of introduction to this molecular identification of genera and species of cyanobacteria. So it's something for the end of the year.
Anyone else involved already in organizing training courses of anything?
In our university, we have a course based on biodiversity and biotechnological potential of microalgae. I think it will open in October. 2 weeks intensive course.
And you'll be involved in that, and doing what kind of thing?
We will try to show the people and teach how to analyze, for example, lipids, fatty acids in gas chromatography from microalgae, how to extract, how to analyze, and how to understand the data. The same for pigments in HPLC, but my point is more related to biotechnological purposes.
Okay.
Yes, thank you. I just wanted to share with some of you, all of you, a project that we are developing. So maybe I can go there.
Yeah, sure.
Thank you. Okay, so this is a European project. I hope that we are well referenced here. Clusters project. So the idea of the project is that we want to help all the European coastal regions to develop their economy, blue economy. And so for that, we are developing a platform that will be launched after summer this year, and the name will be Blue Bioeconomy Hub. And so for us, the idea is that all of you, all the actors within Blue Bioeconomy could join the platform and create a profile. And then the idea is that we are going to create forums for the different value chains. So there will be a forum for microalgae, bivalves, the different species or bioresources. So the idea is that we are going to animate that, and that we will have different kind of stakeholders. So you will have normally enterprises there, industrials, and all of this. Sorry, but this is not the official. Yeah, the Spanish keyword is not the same. So, ¿Cómo borro para empezar? Retroceso. Yeah. Okay. Es www.blue .eu. Anyways, the platform will be launched after summer, as I said, and is a mix between a LinkedIn, is a social network, but also a place for everyone that it's involved in blue bioeconomy to interact, to try to engage and promote this kind of interaction between industry, academia, and research centers. We are the 10 partners through Europe, and then go to this one, Polmer. Yeah, and I think at the bottom of the page, normally you will have the. But I could share the link with you, and just telling you when the platform will be launched. And so the idea for us is to develop that, and we are going to develop also some B2B sessions there. So it will be more or less like a catalog, a profile for everyone for the users to search for something in particular, and for the people that are, as you, for example, trying to offer some services, just to put the services there. So that's the idea, and we thought that it could be something for a EBB legacy. I mean, just a way to capitalize on all the work that we have been done in EBB. So yeah, I just want to share with you this other European project. It doesn't matter if your country is not part of the consortium, because our main is to help the development in the whole Europe. So, but I will share with you all of that. And as I said, it will be launched after summer. So for you to keep in mind the name of the project. Thank you.
Thank you. That's very interesting. And it is like EMBRC associated to that, or no? No, not at all. So it was individual.
Yeah, it's mostly clusters, maritime clusters. So the coordinator is a cluster in Germany, but that works with maritime actors in the Baltic basin. So we have partners in Sweden, Estonia, Lithuania, Poland, Portugal, France, and that's it. So I think that will be a cool way to try to get this interaction between industry and research. But of course, if you have maybe something already consolidated between the different biobanks, and that you can just share it as something that you can do like harmonized for the users, I think that will be very interesting.
Okay, thank you. Very interesting. So where were we? Get training. Anyone else?
Yeah, for this action, for the joint action, you know, this is something that is going out from the ABB project. This is the idea, Ian?
No, I mean, I'm talking about the JDA.
Yes.
Because yes. It does actually mention that it could be development of training activities.
Okay. Yeah, but before we were talking about services, for example.
Yeah, but I think they consider that a training activity is a service.
Okay.
So I think I had a quick read through, and I think that would you know, that could be part of a, you know, that could form a JDA to develop a training offer. But so it seems like there's not many people who do that already. Am I right in saying that?
I think most of us we give this kind of courses, but well, everyone is going, you know, in different ways and doing themselves. So maybe it's the time to do something all together.
Because I mean, we in Roscoff we were also thinking about doing a training course on aquariology, you know, husbandry of animals and stuff like that, which, as far as I know, doesn't exist yet. But if you, I don't know quite how that would work though. If you had a training activity as a joint development, that would mean you would do the same training activity in different countries, or you would just have it in one country, or going in between countries, or. And I'm not sure what you would need to do to develop. You know, what budget would you need to do to develop a training activity? I think there's, you could look to use innovative training tools and develop webinars and things like that. But so that, I mean, that would be a possibility. But any comments on the macroalgae, for example?
Yeah, well, with the seaweeds. Yeah, I don't know how much potential we have in EMBRC, but there are some initiatives in Europe. We have been working on one of that, which is to incorporate seaweeds in the biobanks. I mean, just not only laminaria for outdoor cultivation, you know, or offshore cultivation, just trying to keep material from seaweeds that can be useful for other possibilities for cultivation, you know. So just could be something like establish the tools to keep seaweed material in the collections as a service.
Yeah, I mean the service would be the strains, the new strains. I mean, I was wondering whether, because it seems like I'm involved in a few of these seaweed kind of forums and stuff, and it seems like what it's going towards is that to do culture in the sea of seaweed. The idea is not to put a strain which comes from anywhere. And so what it looks like, it's going towards having regional biobanks specifically with regional strains, which would then provide the strains to it wouldn't necessarily how do you call it? Because there needs to be, for farmers, they would eventually need to have a big volumes of seed material. And I think the idea is not necessarily the biobanks that do the big volume. There would be a regional center for doing the big volume, but the biobank would maintain the strains and
species. And just to establish protocols for the different interesting species from every locality. I mean, if we establish the protocols for our collection, but you will have also for your collection, and it could be interesting.
So maybe we can talk a bit more, and I don't know if you would know, is anyone else here interested on seaweeds? No. I thought you had a PhD student on seaweeds. It would have to be. You would have to be in the. I mean, the idea would be then to have the collection in your place, which you would distribute and stuff like that. So, but yeah, I mean, if you're interested, Who else in the EMBRC would be an obvious people to contact for seaweed? I mean, it would have been Oban, but they're not in EMBRC anymore. But Germany, no, they don't need us.
Yeah, but for example, people in Sweden, because Kristineberg is in, and I don't know. Yeah, Sophie.
But as far as I know, they don't have a culture collection, because it has to be people who are then going to provide as a service. So, okay. And what about the fungi idea? And it could also, I mean, there's many other groups of microorganisms which I think are underrepresented in collections. Fungi was an idea, but it could also be other things. I mean, I think you would certainly have a better chance if it was new to all of EMBRC, probably.
Because I'm thinking, for example, using genetic tools for bioprospecting. I don't know if some of you already use these kind of services or have these kind of services.
Genetic tools for bioprospecting. So what exactly are you thinking of?
For example, searching genome, some genes related to a specific
Yeah, as far as I know, that's not offered as a service.
Maybe we can try to
yeah, I mean that would be something interesting to look at. I mean, that's not so much a biological resource center issue. It's more of the bioinformatics platform issue. But it would be interesting. I think I could talk to a few people about that. I mean, I would have no idea how to do it. I'm not a bioinformatician.
Me neither, just one idea.
So, fungi? No? So they appear by accident.
Yeah.
It makes it easy then.
Sometimes they are studying that fungi like they are good for antibiotics or something like that. Because
and that's why I think it would be quite easy to sell a project like that, because they are really interesting for. So we've never really worked on fungi. We have a few strains, but again they were just by accident.
Yeah.
But I think it could be interesting to do a concerted effort around a few different places. And I mean, are methods for doing that fairly standard and established, or is there some method development to do? Or
I guess what we'll do here is to talk to the fungi guy in Marbach.
The fungi guy.
Yeah, sorry, but I presented it here today because I knew it was quite new for many. So every time we present the fungi activity that we have got, it always makes a good response. People get interested, and we have ongoing projects in Norway this summer. That's why Fungi Guy is not here, because he's out in the field, going all the way in the fjords from the south of Norway and up north. So we have a large activity on that, and we know it's interesting. So for us, I guess we're positive. So I don't know the setup of a project like that. So But we will be.
Yeah, but I mean, we
it.
can talk
Yeah.
about that.
Yeah.
We've got until September to do the
Yeah,
project, but
exactly.
I mean, is that something you would be interested in as well or not? Yes.
And I know that there has also been this focus on using seaweed and different. So you know that it's a substrate also for some of the marine fungi. So it could be interesting to have a closer look if it's going to be used for cow feed or whatever. You need to kind of deal with a lot of the issues regarding the seaweed, and I guess that the marine fungi could play an important role in a total overview of things like this.
Yeah, I mean, that could be used as justification. We can't do research on that because that's not developing a service, but the thing would be, isolating new cultures.
Again, I don't know if some of you already offer this kind of service, but for example, established co-cultures, you give this kind of service?
Yeah, no. And certainly microbiome things is a potential. I actually remember now I had another idea, which was providing what are they called, bulk community. No, what are they called when you
cultures?
No, when you make a synthetic mix, a mock community. That's the thing. Mock community, because we actually worked a bit with Emobon, the project, the big EMBRC project, because they wanted some mock community samples to use as a standard for when they're doing extraction and sequencing. And I think there's potential for, he's frowning because it's a bit of a pain to do, but I think that would potentially be a service which could be of interest. So you provide mock communities either as a mix. So you have a standard mix with certain species and certain quantities, or you could extract the DNA from each one and mix the DNA, for example, and use that as a standard for for sequencing activities. And I think there is some research and development to do in that, to check, to what extent, if you make 10 batches of a mock community, would it give the same results for, is that kind of thing. So that's a possibility. But in terms of microbiome, like mixed samples, or I don't know yet whether there is really a demand for that, because the idea is to do a service for that there is a demand for, but I mean that's kind of linked to a mock community service. That could be something we could think about. Okay, well, I can see it's all getting a bit slower, so I think we'll wrap up a little bit early. I think I will try and do a short report on this and put a few points in. Just to finish up, let's talk a little bit about the future of this working group. Does anybody have an opinion on, for a start, is this working group useful? Is it interesting? How should we go about things in the future? Should we set up some? I think what would be nice is to try and have a meeting once a year and organize it a bit more in advance, so you give people more of a chance to be there. I think the one thing I'm not really sure about is whether because for most of us here, it was, we had the funding via EBB for this meeting, but obviously, if for next year, that wouldn't be the case. By the way, we already have the location. It's in Coimbra because they have much better meat and you can party all week apparently. Or what was that the selling point? Something like that. But I don't know whether EMBRC would be able to fund things like that. I'm looking at Arno, but
EMBRC, I think, thinks that what we have in mind is whether meetings in visio and not meetings in person, maybe once a year or in. But this, but the idea was to meet. It was not to move everyone in a single place.
Yeah, but I mean, we can obviously have tele meetings on certain issues for certain things, but I'm not interested to do a 2-day meeting by video conference. It's the interesting thing about these meetings is to be here, and then it's not even necessarily the interesting what we're saying now. It's what you say when you're having coffee and at the bar and stuff like that.
I see the value of this meeting. If you want to talk about a quality system for 2 days, obviously I think it's better to do it like today. But I think about regular meetings. I don't know, maybe not
for
once
start,
a year,
is there
but are you interesting to have more meetings? I don't know which frequency, but more to talk about technical issues or
no, but I think personally, I think that for certain technical issues, you could have video specific technical issues. You could have maybe video meetings, but I think it would be quite nice to aim to have one meeting a year in person where you have different subjects. And another question is, should we look to have like a meeting like this where you talk about lots of different things, or it would also be interesting, I think, to have like a real workshop where you go and do something practical together, like cytometry, or go on a sampling trip, or something more hands-on. Sorry? Do you know if there is a EMBRC Community Days next year or this year?
Yeah, I think it's planned for the next year.
Do you know where?
No.
Because that is also a possibility, is that you could tack it on to EMBRC, do it a day or two before.
Yeah, if there is an EMBRC days, for sure we have time to organize the working group in the same event, and I will just find out when it is, where it is.
In terms of funding, because I think I remember now I asked Nick, and he basically said that's the national nodes. That's their problem. They should fund you if you want to go. I don't know in France if there would be a budget for us to come to a meeting like this, but that was his response at least. But I think if there was a EMBRC Community Days, then the national nodes would fund our participation, would they? Presumably. So that might be an idea. But so any comments on the kind of future of this working group?
We are the last ones to join you here, but we are looking the activities you have been doing during these years, and through the web page and the documents and so. And I think it's quite interesting. The topic of the Nagoya Protocol and the ABS still has a lot of open things. And I think it's quite interesting if people who is working on that regularly can join and go ahead with the new ideas, new problems, and new solutions for that. So I think it's quite nice to to see the possibility to go ahead with the work group and these ideas and well this question maybe try to organize one meeting a year, just trying to update all this information and the activities that we already make in our collections and try to bring new people in the idea, because people is asking us about all these politics and ABS and Nagoya Protocol, so maybe...
Yeah, I think for next year, trying to link it to the EMBRC community days would be probably a good idea. Also, another question is, would there be an interest for specific things? What I was thinking, I didn't ask Martin, but would there be an interest within this working group to organize specific practical training sessions? And I was thinking of flow cytometry, because there was a few people who said they would like to know more about flow cytometry, and that's something we could potentially organize within this. Would be, it might only be 2 or 3 participants, but go somewhere where they are specialized in a technique and do a couple of days of you have a comment? Yeah, but that makes it a big thing. I'm thinking more specifically on a technical, and I'm thinking flow cytometry, but I'm sure there are other issues. I mean, could be something, for example, on large-scale culture, which you could organize a couple of days, and that might be something which would be interesting to people, and really a practical, hands-on. Let's for a couple of days. Let's really get into this. And I'm not saying for this. The idea is not to have 20 participants. It's 2 or 3 people who are really interested, and obviously the funding. I don't think we would get funding via EMBRC for that. I can ask, but so the funding might be an issue. You would probably have to fund yourself to get there if that was the case. But I think that could be and if that is of interest, what we would need to know is what kind of techniques or things you want to have some training in, and what kind of techniques you can offer some training in. So I think for that, I shouldn't say this because I probably never get around to doing it, but I would try to put in place, I don't know, a Google Doc or a Drive or something where we can, I can put, if I manage to do some notes of this meeting and have somewhere where we can just exchange on, you could put in that you're interested in certain thing and that you can offer certain thing, and I think that could be quite a nice. Actually, I say the funding, but Arnold, do you know if I think there is funding in EMBRC for staff exchange?
I have no more information since yesterday, but I think so. I just need to find out.
Yeah, so within the Assemble project, there was a staff exchange program where you could make just a 1-page application, and you would get your travel and subsistence paid for I think it was up to a few weeks or something. And I think EMBRC has put in place a similar thing now. Yeah, they've been talking about it for ages, and I think it's in place, so that could fund that kind of training activity, I think. And what would be good is, because we quite often have people say, can I come and learn something for a week? It'd be good if the person who's doing the training takes the time to do that. It's better if there are at least 2 or 3 or 4 people and try and do it all together. And so I'll try and put in place some kind of drive or something to do that. Another comment is this working group. I don't quite know how I got elected to be in charge of it. I never volunteered. So, if there's anyone who is motivated to help out, and I'll be very happy. Maybe we should actually have elections every year and change the. So yeah, that's a great idea. So next year we'll have elections for who becomes the new head of this working group. I vote Sergio.
If there is regular video conference, I think I can organize them for you, but you to create the content, the topics, the discussions needs to be. So yeah. I cannot suggest your topic.
Yeah, and well, thanks for the offer, but the thing is, I like the idea of having all these exchanges and video conference meetings and stuff, but I know that when I get back, I'll have a million other things to do and might not get around to it. But let's see. So in any case, any encouragement or help would be welcome on that aspect if you think it's a useful thing to do. So with that, I think we should maybe wrap up. I would like to take the opportunity to thank the organizers, all of you, very much. To thank our IT guys as well. Round of applause. And really for your hosting of this, you've made us feel very welcome, and it's been a very nice experience, I think, for all of us. So thanks very much for that. And hopefully we'll be back to collaborate soon on different things. So, and thank you all for coming, obviously. And I think it's aperitif time. So do we have a plan for tonight? So I'll hand over to the social manager.
We're going to watch the football match at 9 in one bar close to the Las Canteras, and then we will take something until 4. He has to take a taxi at 4, so we have to stay with him until 4. So this is the plan. It's okay.